I'll hand over to Dr. Chris Harrington from the SAS Trace Element Laboratory in Guildford. And thank you for your talk today, Chris.
Okay, thanks a lot, Mark. Good afternoon, everybody. I work down in Guildford on the Surrey Research Park in the SAS Trace Element Lab. So that's a regional laboratory for, in our case, for the analysis of trace elements. So we've got lots of very expensive instrumentation we use, and also we get to see quite a lot of unusual cases to help us with the clinical interpretation around obviously trace elements. We're also home to the main EQA scheme for trace elements that's used in the UK, but also internationally. I'm going to be talking today about my sort of experiences in doing research and development in we're a district general hospital, is where I'm based, and obviously around clinical trace elements. So I thought I'd start by sort of outlining what I think are the kind of key ingredients to try and get research together. Obviously, there's lots of components to get working, to bring together to get a research project, and then hopefully to get some sort of successful outcome. But obviously, you've got to start with an idea or a hypothesis to test. Obviously, research is based around testing hypotheses. And there might be a lot of drivers for this, might be some new, so this is all around really the diagnostic area, monitoring and diagnosis, which is sort of where we work. And it might be because there's a new approach, a new analytical approach, or a new clinical approach. There could be new guidelines or clinical pathways that you need to provide information for clinicians. Usually it has to involve some sort of clinical input, and obviously ultimately we're trying to benefit the patients in some way. So to do that, we need available instrumentation. Now that could be instrumentation that you use routinely, and you can create some time on there, or you might have to find some funding to purchase suitable equipment to do what you're trying to do. And obviously with anything you need some consumables. So immediately you've generated a requirement there to get some funding off somebody. So that's obviously initially in the first stages of the project where you've got to move to. You need somewhere suitable to do it. And it's often good to include collaboration. So that could be with another university or hospital, or often you want to be collaborating with clinicians, and ideal collaborations are where people bring different things to the table and try and answer the question. But we've also in the past we've also benefited from working quite closely with instrument manufacturers, and they've got a vested interest really. They want to see people use their instruments. It gives them sort of data and material that they can use in their marketing, and they can also profile you so that other people in the area that you're working in can see that these instruments do work and are useful in the measurements. Obviously you need personnel to do that and a level of training, and that really depends on what part of the project they're involved in. So it could be right from trainees or visitors to people with more experience in research and development, depending on how complex the project is. Coming into funding, obviously what you want to do at the start is to get some seed funding. So your department could help you with that, or you might have a charity at your hospital trust, or I've often found that professional bodies are really good at this. They offer small startup grants. They also offer travel funding, so that could be either for you to visit somebody or to somebody to visit you. And if somebody comes and visits you, obviously they may bring with some consumable money as well with them. And obviously, if it's the right sort of person and fit with the project, they're going to have some experience they bring to the table as well. You could generate some commercial work to fund it, and you could speak nicely to your finance director, and hopefully they'll let you use some of that money for the consumables. But the ultimate idea really is to get some sort of preliminary data so that you can apply for a large grant. And it's often quite good if you can get collaborators who are more experienced or got an established track record, because you can leverage their experience and publication record to really help with your application. But ultimately you need some output, and this is really a kind of a feedback loop. If you can get some outputs from a project, you can show a track record. If you can show a track record, then people are probably more likely to sort of go with you, because you've shown that you can actually go from that idea to actually producing something. And obviously these are the sort of standard outputs, deliverables that you get. It's often useful also to think about trade magazines in your particular area, or magazines or journals around your professional bodies, because that you're going to get a real targeted audience there. And really, I suppose what you're trying to come up with for some of these things is a sort of best practice guide, so there's true patient benefit. And then obviously you need some luck and a lot of commitment so that you don't sort of fall at the first hurdle, because it's not necessarily that easy to do these things. So I thought I'd talk about some work that I started quite a long time ago now, 2011, and this was all around metal and metal hip replacement devices. So this was a collaboration that sort of kicked off then between myself and my predecessor, Dr. Andrew Taylor. We were based at the Trace Element Lab, and we also used in this collaboration some of our sort of knowledge of the EQA scheme. And this sort of was instigated by some orthopedic surgeons in the Northern Retrieval Registry in Newcastle. And we also worked on this particular project with the National Measurement Institute, this LGC, who were based in Editon, and in Teddington, in particular, Dr. Steve Ellison. And we had a number of clinical scientists, trainee products working on this. So I'm sure you've all come across hip replacements. They're pretty common now to alleviate sort of pain and help people with their mobility problems. These particular type of hip are constructed from these so-called metal on metal hips are constructed from cobalt, chromium and molybdenum, and they were introduced quite a long time ago now. And they were really targeted at younger active patients because they kind of replaced or worked alongside plastic hip replacements. And so obviously with the younger person, they're going to have a hip replacement for a longer time, and they may be more active. So these were seen as being a good use because they obviously they were thought to last longer, so that people wouldn't have so many second hip replacement through sort of their active lifestyle. There's two systems that are commonly used. There's this total hip replacement, and that's where the head of the femur is removed and a femoral stem is placed in the top of the femur. And that's made out of titanium, and that sort of fits to this point here where we have a ball and a cup. And these are made out of this cobalt, chromium and molybdenum alloy. And then the other sort of modality is to not take the top of the femur off, but to basically resurface it. And in this case, we have just a ball and a cup. And these were introduced quite a long time ago, but it became quite apparent in this orthopedic's mind that his group of patients were coming back with pain, issues around pain, problems with mobility when they had these implants in for a few years, and also this sort of distinctive clicking noise. And when they did some imaging on this, it became clear that there was quite a big problem, particularly with inflammation. And so they opened these hips up again, and they found some necrotic material and sort of lots of signs of inflammation around the hip joint. And they worked quite a while on that, and they approached us to get involved in this work. But obviously this was quite a big issue at the time, and with some of these hips were actually removed from the market. So the larger hips tended to cause quite a lot of problems, and it was all to do with wear around where the cup met the ball here, and that if this angle here wasn't quite right, and also a lot to do with the size of it, it would generate what are called wear particles, and that would lead to these inflammation responses, necrosis, and then loosening of the bone, and ultimately that would lead to the patient having to have another hip replacement, and obviously quite a lot of discomfort as well. So back in 2010, the regulator got involved. The MHRA had issued this advisory notice, which was that hospitals should put in place systems to follow up of patients implanted with these hips, where they would look at measuring the cobalt and chromium levels. And then depending on those levels, they would then go on to have MRI or ultrasound to do further investigations, further imaging. So in this case, cobalt and chromium, which is something that we did measure prior to this in the lab, was really used as a screening method. And back in 2010, when this started, I think in 2009, we were doing about 4 or 5 of these a year, and that went up to now. So we're now in 4 or 5,000 a year. So it went almost exponential overnight. Well, one of the things back in the early days they were interested in was when we're measuring this as a screening. So this is blood cobalt levels here, and this is the surface wear of these components. Was blood lead a good representation of the amount of wear there was? So we were working with this retrieval centre, so they were taking the hips back out, and then they were doing techniques around tribiology where they look at the surface and measure the irregularities very accurately, and they could work out how much wear material had come off, and then when they compared that to the blood cobalt, there was a very good relationship between the two. So that gave everyone confidence that it was a useful screening tool. And in fact, when they looked at their populations much more closely, because they had a large sort of population of patients under their care, they found that within the range that was the cutoff, this 7 ppb or micrograms per litre of cobalt or chromium, it did have high sensitivity and specificity for abnormal wear. And as this increased, we saw an increased number of problems with the hip leading up to osteolysis, which is this breakdown and loosening of the hip, which ultimately leads potentially to the hip having to be replaced. So at the time, so this was quite a while ago now, there was the clinicians were a bit wary of these analysis, and it became clear that because this was going to be an annual test, so the MHRA guidelines said for the first 5 years they needed an annual test, and then the local team could then decide what they were doing ongoing after those 5 years. How they were concerned that they were concerned that any changes they would see would be not to do with the hip replacement, but could be to do with the measurement. So, well, it was quite lucky that our EQA scheme had included cobalt and chromium in these samples. These were equine blood that we spiked out, and we had 30 participants on this particular scheme. And when we looked back over the data, we could show that the recovery of the spike was very good, and it was extremely precise as well for over the last 12 months for 30 participants. It was 96%. So we published that in BMJ just to sort of reassure people. So that did show that these results were consistent over time. But obviously the labs that were doing this, they were concerned whether these results were actually accurate. So the technique that we use in most laboratories we used at the time, and we still use, is this technique called inductively coupled plasma mass spectrometry. And so this is a technique where a plasma, so that's a gas plasma, is induced. So that's a very, very hot source for the production of iron. So it's very hot, and it contains a lot of gas ions that can convert the blood or serum or urine or whatever goes into it into atoms, and then ultimately into these ions. And then we can measure these ions in the mass spectrometer, and we can measure them at their mass to charge ratio. And that's what we proceeded to do. But the problem with these two elements, they're quite difficult to do. Cobalt is monoisotopic, so it only has a single isotope. So if there was any interference on this, then we wouldn't have a second isotope to kind of check whether the signal was correct. And chromium had significant interferences from carbon. So how do we know the measurement is accurate? Well, we approached the laboratory of the government chemist, and we got involved in a project with them to prepare a certified reference material. So that's effectively what LGC does. It produces accurate methods and standards and materials you could use to validate and show accuracy in methods. And so we spent some time working with them using our skills as an EQA provider to basically prepare a certified reference material, and then they use some of their very high accurate methods based on ICP-MS, but with some more expensive instrumentation to give it a certified value. And we also did some commutability studies against patient samples. That's where we showed basically that equine blood was a suitable replacement to show what it was like in human blood. So that then gave everyone basically a material that they could show their methods worked with. So that kind of progressed to that stage. And then back in 2014, we started getting interested in these nanoparticles that were produced, these wear particles, and what were they actually doing. So nobody knew at that time what the composition was for them, how many there were, or the size of them. And obviously there could be some issue, there could be some some of those particles could be more or less immunogenic than others. So you could basically get some information on which hip types were going to be more problematic than others. So again, I worked with the Northern Retrieval Registry with Dr. David Langton, and I had some colleagues that I'd sort of knew from various conferences working in the food industry. So this was in Technical University of Denmark, and in the food industry, they use nanomaterials as coatings for food packaging, and there was an interest at the time in transfer of those particles. So they'd developed some methods to look at what is a very difficult thing to do, to actually measure the composition, number and size of nanoparticles. And at that time, the EU was very interested because these nanoparticles were getting in the environment, and so they put regulations in place for the measurement of nanomaterials, or regulations for the size of nanomaterials, which meant that everyone had to come up with methods. So I went for a week, travelled over to Copenhagen, which was very nice, and worked in their lab using their established methods, and some samples that were from aspirates around the actual implants. They had very, very high levels of cobalt, chromium and molybdenum in them. We did some work on those, and I'll show you the results from that in a moment. And I realized this was quite a big area. So I looked sort of for a local collaborator, and I came across Professor John Watts at the University of Surrey. And using the income from the CRM production, we managed to get a joint PhD student to sort of look further into this area. So this was a collaboration really between a number of different stakeholders. So the method they were using was this so-called single particle ICP-MS. So again, we're using the inductively coupled plasma mass spectrometer, but in this case, we're using a technique called single particles, and that kind of works like this. So this is a schematic of an inductively coupled plasma. This is an induction coil here, and we have an argon gas that comes through here. And when we induce a magnetic field in here, this produces our plasma, which is very, very hot and got lots of gas ions in it. And then that basically blasts everything apart till it forms atoms and then ions. And then this part here is a mass spectrometer. So this part is under reduced pressure. This is at atmospheric pressure. So we have to go through a very small hole into the mass spectrometer where we separate and detect the ions. So the way that this technique works basically is if we measure this very, very slowly, so we're measuring cobalt and chromium very, very slowly, but if you think of a particle, it has a lot of atoms in it. So when that goes through and gets measured into the mass spectrometer, we get a much bigger peak than if we're measuring smaller particles. We could then use some software that comes with the instrument, so we could look at the size of these peaks, and that gives us an indication of the size of the particle. And then the number of those peaks tells us how many we've got there. So we can basically work out some size distributions, and we're sort of working down at the nanogram per litre range to do this. So this is what we got when we looked at it. So this is the raw trace of chromium and cobalt here, and you can see every one of these peaks is a particle that's gone into the instrument. And when we sort of deconvolute these traces, we can basically work out a size distribution, and it worked out that these nanoparticles were down in this size range here for cobalt and this size range here for chromium. And we basically fitted these results to get this distribution of peaks. In this case for cobalt, we're a bit limited on getting the full distribution because of the limit of detection, because obviously the smaller the peak, we need the power of the detection limit to be able to work it out. So we only see half of the distribution here. Anyway, it gave us a kind of a first look at doing this sort of thing, but it was quite a difficult thing to do. But the model that it uses, so this is basically different materials here, so it's comparing the distribution size here to these industry defined compositions, which are for the different hip replacements that you get, or to whether it was chromium oxide or cobalt oxide. So we use this information to work out this size distribution, but it basically assumes that it's a particle. So the work we did with the University of Surrey was to use electron microscopy to basically look at these things and see if these particles were actually spherical or what shape they were. Now that sounded quite easy when we started off, but as soon as you put any biological material in an SEM, it just throws back a load of basically reflects back all the radiation. You don't see any detail. So we had to develop a lot of purification methods to get to this point, which were based on centrifugation and basically diluting down all the organic stuff that was in there. And eventually we did see some of these peaks here. And if you use transmission electron microscopy, you can actually measure the wavelengths that come off here, and we could see the composition, including molybdenum, cobalt. There's also peaks for iron, nickel that were present in there. Ultimately, the student got a PhD out of that, but it turned out to be a lot more complicated than we anticipated, and that was really as far as that part of the work went. The other thing we were interested in was, although it's pretty well defined what the material composition is, when you actually look at patient samples, you see a much greater buildup of cobalt. So you see more cobalt than you do chromium compared to the composition of the original material. We were interested in why that might be. So we thought, well, we look at how these different metals are bound up to the different serum proteins. And in this case, we used anion exchange chromatography. So that's a method of separating out the proteins. And we then coupled that up to our ICP-MS to detect the different metals. So this was some of the work that we did in Copenhagen. This was for 7 serum samples from these patients. And you could see that, unsurprisingly, really, because there are binding sites for cobalt on albumin, there's a much higher concentration of cobalt bound to albumin than there is chromium. And albumin is such a high concentration that effectively we're seeing retention of cobalt bound up to albumin. We also see chromium bound to transferrin, which is quite interesting because transferrin is involved in production of blood and does end up going back to the bone marrow. So it was interesting to speculate what effect what transferrin might have with chromium bound to it. But it did answer this question we had, and it turned out when we looked in the hip fluids there was very high levels of albumin, and that was later used by the people up in Newcastle to develop some further assays that I'll go on to tell you about in a moment. Once I've gone through the last part of this, or the second to last part of this collaboration, was started in 2016, and that's when Dave from Northern Retrieval Registry came and said, well, we're interested in measuring titanium because we think there's something going on there with that stem that fits into the back of that ball. We think there's some wear going on there. Now, titanium's pretty difficult metal to measure, as it turns out. So we approached Thermo Fisher Scientific, and we developed a collaboration with them. We also worked with some of our clinical scientist trainees. So this was a collaboration between us and Thermo Scientific, where they actually ended up loaning us an instrument for a couple of years free of charge, which was very nice of them. So in this schematic here, this is where that's occurring between this stem and where it meets the sort of the top of the hip replacement. And they could use their tribiology technique to show that there was significant wear here. Well, what they were seeing in the patients was they were seeing soft tissue damage when they had less particles present in the hip fluid, and they wondered and speculated whether it was titanium. And that was compared to where we have bearing surface. So this is the top, this is this part here. This is how it basically was wearing. They were getting a lot less soft tissue damage, but larger particles. So they wanted to know, well, how much titanium is present in samples? Well, titanium's not very easy to measure, as it turns out, because the main isotope in ICP-MS, so this is the most abundant. So if you measure this, you'll get the biggest signal for titanium because most of the titanium is present as this particular form of the metal at this, and it occurs at mass charge ratio 48. And unfortunately, that's the same mass charge ratio as calcium. And obviously, we've got very large, in comparison to titanium, large amounts of calcium present in the in samples. So effectively, we can't measure it at the most abundant isotope. We could do it if we had a high resolution instrument like they do at LGC, but unfortunately we don't. They're very expensive and we don't really have the capability to run it. And at the time, there was a new technique which we have now got in the lab, but at the time we didn't, which is this MSMS. So this is a technique where you react the titanium with, in this case, ammonia gas to form this cluster ion, which occurs at about 150. So it basically moves the titanium isotope here at 48 up the scale away from calcium, because calcium doesn't react with the ammonia. But that was expensive at the time, and it also had a lot of health and safety issues about having ammonia in the lab. It turned out we couldn't really do that. But after speaking to Thermo Fisher, they said, well, have you thought of using this technique called optical emission spectroscopy? So in this case, this still has this plasma, this very hot, basically source of producing ions, but instead of looking at what's coming off there in terms of the isotopes that come off, we're looking at the light that's generated from this. So each of these atoms that goes in there will have a specific emission spectra, and you can measure the amount of emission that comes off there, and you can measure the amount of titanium. So that's the way we did it, and we didn't really have any experience of this when we started, so it took quite a long time to get it to work. But when we did, and it had never been done in human samples before, we did compare it to the methods they were using, these triple quadrupole methods here using the ammonia, and we saw pretty good agreement. So we're seeing good linear agreement there. And when we look at the difference between them, it's within plus or minus 2 SD, which is how we kind of work to establish that things are in agreement. So we got good agreement within plus or minus 2 SD. And then we went off and started looking at a lot of samples to try and work out, what was the distribution in these patients, what sort of levels were there. We also had to identify what sample matrix to actually look at as well. And we came upon using serum, which was reflective of what was in there. But we also did look at whole blood and hip fluid, and we did end up publishing this as a method. So this was a new method for the routine measurement of titanium. There hadn't been any methods to do it in these samples prior to this. And also, as part of the calibration, we did a round robin study with people from various places, basically the Health and Safety Executive, LGC, and the MRC in Cambridge. And we did a round robin where we were the only ones using this method. They used different ICP-MS methods, and we got very good agreement with these other methods. So we had a method to look at titanium. So that sort of brings me near to the end of the talk now. So going on from that, the Northern Retrieval people, Dave Langton, they set up this company, Xplant. And this is an orthogenomic biotechnology company, where they provide this test, which we provide the analysis for. So what they're trying to do here is come up with a risk profile. So they're using, so it turns out that a lot of patients in the UK carry genes that give them an increased susceptibility to inflammation as a result of having these nanoparticles in them. So they're using, this was stuff that they did with the University of Newcastle. They're looking at next generation sequencing to identify patients who've got this particular type of inflammation marker. And using that together with our results, they can use this orthotype algorithm to give them some sort of risk. So they can provide a risk profile to the patients prior to giving them the implant. That's the idea. And then they've also come up with this other test using titanium. So using the measurement of titanium that we had to look at implants. And this has got a sensitivity and specificity indicated here. And again, this could identify abnormal wear. So titanium is not just used in hips, it's also used in knee. So there seems to be quite a lot of issues also around knees at the moment. So patients can, with advice from their clinicians, can use these tests to sort of identify if they're going to get any risk outcomes from it. So just what did we do going back from 2011? What did we produce? Well, we did produce quite a significant income into the sort of tens, if not hundreds of thousands of pounds as a result of the work that we've done in hip replacements. We also developed that certified reference material that's still available, so people can use that to establish the accuracy of their methods. We got that instrument. That was saved us £60,000. Obviously, we had to give it back, but we'd shown the proof of principle there. We also did a lot of training, a number of clinical scientist projects for Royal Surrey. We also had the PhD and MSc students from the University of Surrey. Also King's College London visited, and we had some overseas visitors who worked with us on this project. We produced 5 peer-reviewed research papers, 12 invited presentations, and 4 conference posters. And it led on to this sort of new availability of a test for patients and clinicians, so that they could basically identify if they're at risk from using these particular these particular hip replacements. So hopefully that's shown you what you can achieve if you have the opportunity. Happy to take any questions or respond to any inquiries, etc.